is Rachel Davis, and my project is engineering biodegradable flame retardant polymers. And I go to school at Smithtown High School East, and I worked in the Garcia MRSEC program with Dr. Miriam Rafailovich. The reason that I got involved with flame retardant materials was because 5 years ago my house burned down. It was really a traumatic experience. Everyone was home, and just hearing, when you hear the smoke alarm go off, the first thing you hear is, the first thing that you think is, oh, I don't have to go, it's just the smoke alarm. Mom probably burned something while she was cooking. And then when I heard that it actually happened to me, and that this was for real, when my dad was running through the house and telling us to get out, that it replays over and over again. It really does affect how you see life, and it makes you want to go out and prevent this from happening to other people in the future. Afterwards, my father and I joined the local fire department. We both volunteer at the Nisqually Volunteer Fire Department. And after seeing what happens to all these different people with the house burning down and the effects that fire has in everyone's lives, I wanted to add to preventing this from happening to other people, and I decided to research flame retardant polymers. And these plastics resist the effects of fire and slow down the rate at which fire progresses in these polymers. When I was doing my research, one of the major setbacks was that actually the biggest part of my experiment, what's called a braid bender or a plasticorder, what mixes the polymers, broke. And that's the most important part of my experiment. So for the first 3 months of my experiment, I couldn't actually do research. I was looking at the composition of my materials, but I wasn't actually able to create any material. So that was definitely a setback. But afterwards, I went back every day during school year, and I made it happen. Being in the Intel competition is just so incredible. When I heard that it happened to me, I just couldn't believe it. It's so surreal, and it still hasn't settled in yet that this is actually happening to me.
My name's Julie Carrar. The title of my project was The Impact of Compressive Misfit Strain on Improper Ferroelectricity in Lead Titanate, Strontium Titanate Superlattices. I really got into my project because I've always been fascinated by patterns, and that kind of led into my interest in crystallography because unit cells are basically patterns, repeating arrangements of atoms throughout a crystal. And what's neat about ferroelectrics is that their ferroelectricity, their electrical properties, are specifically related to their internal crystal structure. So if you modify the structure at all, you modify the electric polarization. So I dealt with a specific kind of ferroelectricity called improper ferroelectricity that results when you put two different kinds of materials together, lead titanate and strontium titanate, and kind of alternate them, layers. And so this improper ferroelectricity creates a material that's temperature independent, which it might be interesting for far in the future for applications that these ferroelectrics can be used in capacitors, which are used in sometimes in electric motors and cell phones. So if you use these improperly ferroelectric materials in something that changes with temperature, so motors obviously heat up as they're used, then the efficiency of these materials won't decrease as the temperature increases. I was one of those people who loved arranging my Chex Mix things and poker chips that you would, not actual poker chips, but anything you'd find around the house, I would arrange them in specific patterns while I was playing poker. So I would look in nature too and find all of these different patterns, and I didn't realize that there was actually some science that was related to that. So when I started learning about crystallography, I was like, wow, this is what I want to do with the rest of my life. I want to learn more about these patterns that you actually see in these microscopic crystals, and see how those patterns affect what we actually see.
My name is Neil Mehta, and like Julie said, she's really interested in patterns and crystals. I was really interested in how you can have a bunch of cells that you can throw them together almost. That's what I thought of as a child, almost like chaos. But somehow they beautifully came together and formed this magic organ, what we call the brain, with all its intricate networks and connections that seem so random to us, but when they've been examined, they seem to be perfectly ordered. And I found that really fascinating. And then what I found even more fascinating was how just a slight tweak in those connections, a slight tweak in that order, and a slight change in the order of how things are put together in the brain could lead to some devastating psychiatric disorders. My project is titled Co-Restoration of Type 3 Neuronal 1 Back Signaling Implications for Schizophrenia. And what I basically did was I looked at a mutation of the Neuronal 1 gene in a protein in one of its isoforms called Type 3 Neuronal 1. And what I found was that there are decreased baseline levels and overall levels of two signaling pathways that occur from this protein. And outputs of these signaling pathways are actually implicated in behaviors such as working memory and executive function and learning and other cognitive features that make us human. And so a reduction in those abilities has been hallmark in schizophrenia and other psychiatric disorders. And the mutation I studied was actually statistically linked to schizophrenia, so I found that really interesting. But then I was also wondering, well, if we have this mutation in a protein that has overall reduced capabilities of functioning, well, what can we do about this? And I read a really cool article in the New York Times about a technique called optogenetics, in which you express a protein derived from bacteria that opens when hit by a certain wavelength of light and allows for positively charged ions or cations to flow inside cells. And I remember reading, like a year prior to reading that article, in a journal that tested depolarization of neurons and saw that depolarization can alter the signaling pathways of type 3 Neuronal 1. So after reading that New York Times article, I was thinking, well, could I possibly apply this technique to try and recover the function that was lost as a result of this mutation? And I found that it worked beautifully, and that this optogenetic technique, in fact, restored protein function to less than 50% below than the original protein, compared to about 55% below. So what makes us human is our ability to feel empathy and sympathy for one another in times of distress, and our ability to communicate these emotions with one another, and with that, our ability to learn about one another and learn about what goes on in our world, understand them, and take that information and apply it to make our lives better. And that really is what separates us from, I guess, other species and makes us human.
My name is Savina Kim, and I'm also studying neurobiology like Neil. My project is titled The Cognitive Deficits in Neuropsychiatric Disorders, a schizophrenia model associated with neuronal alpha-7 nicotinic acetylcholine receptor localization. And so if you look at postmortem studies of, let's say, people with neuropsychiatric disorders like schizophrenia, you see that they have a dramatic decrease in these necessary neurotransmitter receptors in their brains. So I was trying to understand, okay, why is this happening in these kinds of patients? So I was looking at these receptors on like a molecular level and trying to understand the actual details of how these receptors become activated and functional. Hopefully later on, when you actually understand the specific signaling pathways and molecular mechanisms by which these receptors localize, then you can actually create more drug therapies to actually target these specific signaling pathways rather than trying to, let's say, alter the actual genetic link behind it. I started my research at Stony Brook 2 summers ago, and just as Neil said, the brain is pretty much this huge mystery to us. And ironically, it's our most important organ, yet we know the least amount about it. And so that's what initially caught my attention. And I think my biggest inspiration was really my younger brother. He's only 10, but he's always been so interested in like the tiny little details. Like, he's the type of kid that I saw reading a MCAT 7th edition book when really he thought it was for 7th graders. And he thinks like SAT represents science and technology. And he just has this just pure and innocent like curiosity for these really smaller details, and that's what really caught my attention, because really you have to understand what's exactly going on underneath what you see, and it's really those underlying intricate sort of details in the brain that's really what matters, and that's what really is necessary when you want to continue it on into more of the clinical field. It's trying to understand what exactly you're dealing with. And within this huge brain and these intricate links and this huge mystery, it'd be nice if maybe I can even just understand one little tiny part of that brain and be able to clear up one mystery.
My name is Anna Sato, and I was in the Simons program at Stony Brook University last summer. It's a research program for high school students. My research title was a novel absorptive filtration approach for the removal of radioactive isotopes of iodine and cesium from water. Basically, my project was creating a membrane that used charge attraction to absorb like iodine and cesium ions from water. My project was inspired or motivated when I saw the newscasts and all the videos and pictures from Japan on TV when the earthquake and tsunami occurred and the Fukushima power plant had the explosion and radioactive isotopes were leaked into the water. Actually, at the dinner table when me and my parents were watching TV, and it was really horrifying images and videos of all the destruction that was caused. And from that, I wanted to help in some way, and I thought maybe I could use my filtration experience in the lab and try to remove radioactive isotopes from water. My parents originally thought, there's no way you can remove radioactive substances from water. It's dangerous. You're just a high school student and you have no safety training to handle these radioactive materials. But my professors, Professor Chu and Professor Shao in the chemistry department, always told me to dream big and try to do whatever you want to do. And so I met with them and kind of asked, do you think it's possible to remove radioactive isotopes from water, or handle a substance that would work to to model radioactive isotopes? And we were able to come up with a method that made it possible. The filtration can be applied to so many different fields and markets, and what I did with water filtration and radioactive isotopes was just a little part of a huge field and opportunities that are available. We can't live without water. It's one of our basic needs just to survive and live. So thank you. My name is Anna Sato.
Music and all of my instruments just burst into flame. It's invaluable, and it's can't buy another one. That's
Actually,
exactly
because I play the guitar too, and my guitar was actually okay. It's everything else.
Oh.
I know. That's something.
So you lost everything except the guitar.
No. And then
You
I
still have
went
the
to
guitar?
my first structural fire actually was 2 months ago, and that's the first thing the guys grabbed actually in their house. I was really, your house is on fire, and you're grabbing your guitar. But then when you think of it, it's okay, I guess whatever. You would grab your instrument, right?
Exactly. Yeah.
Yeah, whatever. You think of what matters to you. I thought I was going right back in, so I'm I'm not gonna grab anything. That's I'm not.
Yeah, because especially with instruments like mine, I feel like you can't have another one that's exactly the same. Of course, you can buy another one that's similar, but it's a one of a kind instrument because it's all hand done, and you can't just go out and buy another one. Okay, that's the same. It's not, and yes. Flame deterrent keys would be very helpful. Everyone would want that.
Yes, exactly. It would be very helpful.
Yeah.
So do you guys play any sports?
Talk about ukulele.
I play ukulele. I actually brought it to the lab a bunch of times because we did have free time since my major piece of equipment was broken. And a bunch of us would just hang out in room 210. It was our hangout spot, and I used to bring my ukulele into the lab, and I wasn't supposed to though, but I did. And I would just play in 210, and we'd go out to go get Dunkin' Donuts, and I just bring
Did
my
Falvich
ukulele.
play with you?
No.
Oh. That would be funny.
No, she did watch though. And it was very funny, and sometimes because it was very crowded, there were so many students in the Garcia program, so I would sit in the corner and just play ukulele while waiting for my test to be done.
What about whitewater rafting?
So I'm a whitewater slalom racer, which is
What got you into that?
because my parents were actually national champions. It was pretty cool. They did that when they were in their 20s, when they were in college. But then when they started having children and whatnot, they didn't get into it until my older brother saw all these canoes and kayaks in the garage, and he was like, "Oh, I want to try those out." So my dad was like, "Okay, yeah, sure, let's go to a slalom race." And so eventually, like, he got all of myself and my sister and my younger brother into it too, and it's been really cool. I go like every weekend.
So are you like a professional?
No, I'm not good enough to be
Junior
a
Olympics?
professional.
Yeah.
I haven't gone to JOs, but
See, there's an acronym for that?
yeah. My twin sister went to JOs, and she had a lot of fun with that. But like, I've been to age group nationals, and I got second in my category.
Wow.
That's impressive. But it's like it's a really tiny sport in the U.S.
But we win
but
everything?
in Europe, then that was a couple like when my parents were racing. That
It's like
the
10
U.S.
years
was
ago.
done like 20 years ago. My parents are about 55 now, so about 30 years ago. But yeah, back then U.S. was winning everything, but now the U.S. has really gotten
Lazy.
yeah, there are not a lot of
McDonald's for
whitewater
that.
slalom racers, so Europe is winning everything in whitewater slalom. But like all the people who make boats and whatnot, they're in Czechoslovakia and all those places. No one makes boats in Europe, in the U.S. anymore.
No,
But
we just use them as cruises.
yeah,
Do you play an instrument?
I played viola back in the day, like a few years ago, and it was pretty interesting experience. Getting bored of the viola, so I started playing violin parts on the viola. Especially, yeah, right?
Yeah.
And hiding that from the conductor was also pretty fun because I was always first chair. So yeah, it was definitely an interesting experience playing viola, and I kind of stopped playing viola when I got into like my research stuff, and I mean I do play from time to time with my little brother, help him practice sometimes, every now and then, but yeah, it's definitely it's fun, it's relaxing, and yeah, I'm sure you guys all play instruments, right?
no,
Research takes up
I
so
think
much
I'm the
time.
only one. I'm the only one who doesn't play an instrument.
Well,
here.
not a classical instrument anymore. Like I used to play cello, but then research is so time consuming. It's understandable, and you might want to rest. You don't have to. What do you play?
I play the cello too, and cello and piano. I like to teach though, because
Yeah,
I like
teach,
to
yeah.
teach other students. I think that's a really fun experience.
It's like you realize, oh wait, I don't know how to explain that. Or like, oh, I didn't know I could actually say it that way, and then they could pick it up and know how to do it.
And then you get
Yeah,
better.
It's really different from just absorbing what your teacher says and like, okay, let's try it now.
And so I like when you get taught by a teacher and all of a sudden you're doing that, and you're like, oh, that's what she meant. I don't know. It's,
Yeah,
it's
yeah.
like a different perspective.
Do y'all play music in your labs?
I don't play
Just me.
No, I mean, do you have music playing?
Oh yes.
Oh yeah, yeah.
My
Radio's always on.
Everyone else plays Mozart the whole time. There's always some kind of Mozart playing.
We had like pop music, like
That's normal.
yeah.
I have Nigerian music playing. It's very interesting.
I listen to like alternative rock, and it's like Mozart's playing all the time. I'm like, okay.
Yeah,
I
like
usually bring my iPod.
you read like the soccer FIFA or whatever score is always like.
Oh
going
my
on and
God.
I think
Which
Last
one?
summer,
That was one of the
last summer I
graduate
spent the
students.
entire first month watching the World Cup. Like 2 summers ago.
Yeah, it's like when you're
That's all we did. We just watched the World Cup, and our lab has had a girl from Switzerland, had a guy from Nigeria, and a few other people that left recently, just from different countries. So they were all like yelling at the screens, and I really don't care about soccer, so I really didn't get into it. So I was like quiet. I'm trying to do my reading, but then they got me into it. And it was actually pretty interesting.
Yeah, it's good when you're waiting for your like samples to get ready or you know waiting for results.
We would usually just eat pizza during that, but okay. Watch TV. TV? Radio? What?
Yeah, like
we
a
just we
computer.
stream it on our computers. Yeah, we've
Yeah.
pretty big screen monitors.
I think we're done.